of virology. As you know, it's our third lecture in this course, and this lecture is grouped only by specific group members. So there is no actually many groups which can be characterized by similar features, but the enteroviruses is one of them. What's the similar between different members of these groups? So first of all, there are a lot of human and animal pathogens. So it's a very big group. Picornaviridae, it's a family of the viruses. As you see in the name, it includes several words. Pico means small. Ribonucleic acids or RNA, picoRNA, and viruses, that means virus. All of these viruses has naked capsid structure. So all of them has no supercapsid. We have many groups of the members of Picornaviridae family. So first of all, enteroviruses. Enteroviruses, it's exactly what we will discuss today. Not all Picornaviridae members are enteroviruses. So in enteroviruses, we will interesting today in just 5 groups. Polio in type 1, 2, and 3. In Coxsackie A viruses, several serotypes, as you see on the slide. Coxsackie B virus, also only 6 serotypes we are interested in. Echoviruses, also several serotypes, and specific enteroviruses, also several serotypes. We are not interested today in rhinoviruses, cardioviruses, arthroviruses, and hepatoviruses. But you can understand from the names of these groups what kind of system this virus could infect. So enteroviruses, of course, you understand, it can infect intestinal system and primary large and small intestine. Rhinoviruses, infection of upper respiratory tract. Cardioviruses, heart infection. Arthroviruses, primary connective tissue infection. And hepatovirus, its only member here is the hepatitis A virus, infection of the liver, hepatocytes. So about hepatitis, we will talk next time. Today, we will focus on enteroviruses only. Only these several groups we will discuss. So first of all, let's talk about general characteristic of this big group. Virion is naked, as I said. As we told, pico is small, means very small in size. As you see, maximum 30 nanometers in diameter. It's very small viruses, one of the most smallest. Capsid has icosahedral symmetry, enclosing a single-strand positive RNA genome inside. So if there is a single-strand positive RNA, means proteins can be synthesized exactly from the RNA of the virus. So it's no need to make an mRNA. So enteroviruses are resistant to very big diapason of environmental factors, especially we're interested in pH. So it's resistant to acidic pH and resistant to alkalinic pH, so can survive in a lot of parts of our digestive system. Also, it's pretty resistant to detergents, mild sewage treatment, and heat. So heating, you know, can kill a lot of viruses, but depend on the time SO means exposure time, and how many degrees exactly. So just common heating, we suggest up to 100 degrees, and it's usually boiling. So short boiling can't kill picornaviruses. Genome is a messenger, most of picornaviruses, yeah. Some of them, of course, will die. Genome is a messenger ribonucleic acid. That's why, I'm sorry, already told about it. Proteins can be synthesized exactly from the RNA of the virus. Naked genome is sufficient for the infections, what it causes. Virus replicates in cytoplasm, but viral RNA is translated into polyprotein, which is then cleaved into enzymatic and structural proteins. Most viruses are cytolytic, means when the virus go out of the cell, it will destroy the cell always. So cytolytic viruses destroy the cells. One more feature. As I said, it's very resistant viruses because the capsid which surround the viral genome very tough. Environmental conditions, common environmental conditions, totally can't destroy it. Our gastrointestinal tract also can't destroy it. Why we're talking about gastrointestinal tract always? Because the main way for transmission is fecal-oral route. So virus come through the mouth and go out through with the feces. Although the virus may initiate infection in gastrointestinal tract, the enteroviruses rarely cause enteric disease. In fact, most infections are usually asymptomatic. Look here, it's a key point. So we call it enteroviruses. It's transferred through the fecal-oral route, but it's very rare cause problems in intestine. It's just the first place for replication. Then, after the primary replication in intestine, the virus can disseminate into different organs. We will talk about each organ and each situation. The best known and most situated picornavirus is poliovirus, which causes polio infections, epidemics, incidents with also paralytic polio. We know about 3 main serotypes of polioviruses, and for polioviruses, all these characteristics are fit. Another one, Coxsackie virus, are named after the town of Coxsackie, New York state, where they were first isolated in the lab, I mean. They are divided into 2 big groups, A and B, on the basis of certain biological and antigenic differences also, and are further subdivided into many serotypes, as you saw on the previous slide, on the basis of additional antigenic differences. So there are group antigens and typical antigens, specific antigens, I would say. The name echovirus, it's another group, is derived from enteric cytopathic human orphan virus, because their disease associated with this agent was not initially observed and still unknown. So we don't know where these viruses are located by many kind of criteria, but by molecular biological data, right now we could understand where is it, but that time it wasn't. That's why orphan. Since 1967, newly isolated enteroviruses have been distinguished numerically. And right now we know also about several serotypes, several important in epidemiology serotypes. There is an electron micrograph of poliovirus. As you see, poliovirus has ecosahedral capsid, and it's a very visible structure on this picture. Very small and primary round. As you see, it's just picture, electron micrograph picture of polio, which isolated from the viral culture. It's not a sample of the sick patient. It's an culture of the viruses. That's why there are so many viruses, pure viruses without cells, without any kind of mucose, etc. Just viruses. So we told about Picornaviridae family, and I gave some examples. Right now, only about enteroviruses. The name of the enteroviruses do not usually cause enteric disease. All of them are replicate and transmitted by the fecal-oral route. Wait a second, please.
برضه انا مش فاهم هي عايزه ايه، انا مش فاهم برضه هي عايزه ايه.
انا قرفت منها بجد، قرفت بجد.
اقفلوا المايكات يا شباب.
Thank you for waiting. So, as I told, this disease transmitted primary by fecal-oral route. It's usually, of course, not a direct transmission, but indirect only. The disease-produced enteroviruses are determined mainly by difference in tissue tropism and the cytolytic capacity of the virus. So the upper respiratory tract, the oropharynx and intestinal tract are the portals of entry for enteroviruses. So the enteroviruses can primarily multiply in mucosal epithelium in oral pharynx and also intestinal tract, intestinal tract. But never, okay, not never, usually not cause symptoms in that organs and tissues. Okay, the virions are impervious to stomach acids, proteases, and the bile, so not very sensitive and can pass through these barriers. Viral replication is initiated in mucosa, as I said, and also lymphoid tissue of the tonsils and pharynx, and the virus later infect M cells and the lymphocytes of the pair patches. Usually enterocytes in the intestinal mucosa, the primary cells for the replication. Primary viremia spreads Wait a second. Я там огромный. Смотри, вот. Вот идентификатор.
57, 31, 89, 29, 53, 46.
Sorry again. So the primary viremia spread the virus to the specific cells, which usually are receptor cells and the primary target. Why I'm talking about the primary target? Because without this replication, usually virus never cause the symptomatic stage of infection, and we perhaps couldn't know about the viral infection. Of course, for the virus, it's not extremely needed. Virus can replicate itself inside all primary cells, but no need to go to the secondary cells, which could be as a nervous tissue, it could be lymph nodes, spleen, liver, and the secondary viremia after the replication usually are totally connected with the symptomatic stage. There is a picture, a scheme, I would say, of the general pathogenesis of enteroviral infection. Here you can observe the target tissue, which are in the bottom. Can you see here the target tissue, skin, muscles, brain, and we can observe primary tissues here. Primary coming through the oral cavity, and the first replication in oropharynx or intestinal tract. Then after that, primary viremia into the blood go spread viruses into the target tissue. In the target tissue can be different. For our today poliovirus, it can be brain. Not exactly the brain, sometimes it's just nervous tissue. And for Coxsackie virus, it's also sometimes happen. It causes symptoms, following symptoms as encephalitic or paralytic diseases. Also, today we will talk about echo and Coxsackie viruses in different groups, and target tissue can be muscles, for example, for Coxsackie A and B virus. So in thorax can cause pleurodynia, and we observe the breathing problem like devil catch the breath, and heart with viral myocarditis and pericarditis. Skin also can be infected, and we know it's as a hand, foot, and mouth disease caused by the enterovirus members. Next time, we will talk about hepatitis A virus, HAV. No need to miss it with HIV. They are different. Target tissue here is the liver and the primary hepatocytes, and of course cause hepatitis during the replication. As we know, it's a cytolytic virus which destroy the cells and destroy the hepatocytes, provide the hepatitis symptoms. But hepatitis A, fortunately, not very dangerous hepatitis, and the primary could be very well treated. I'm sorry. The mechanism of disease for enterovirus is pretty similar. As I said, comes in the primary organ replication, then transmit through the bloodstream into the second target tissue. If a patient has no serum antibody, enteroviruses spread by viremia to a cell receptor, as we know, which we know as a target tissue. If a patient has antibodies inside the serum, so the replication will be limited in the primary organs, in the primary cells. You know, the cells of upper respiratory tract and intestine. Different picornaviruses has different receptors for target tissue, but all of them has same primary receptors. The infected target tissue determine the subsequent disease. So different target tissue, different disease. Very logic, I would say. Okay. Viral, rather the immune pathologic effect, are usually responsible for causing disease. Secretory antibody response is transitory but can't prevent the initial infection. What does it mean? When the virus comes into the primary organ, and you know it's a epithelial cells of the upper respiratory tract or epithelium of the intestine, tract, if there are secretory immunoglobulin class A are present, we can talk about limiting of the viral disease inside this specific tissue. So usually only person who are already recovered from the real infection has secretory immunoglobulins, or a person who were vaccinated by oral vaccine. It's differences. Serum antibody blocks viremic spread to target tissue and preventing disease, usually much more effective than secretory immunoglobulins. Enteroviruses is shed in fishes for a long period. We would say a person who has no symptoms also can be danger, asymptomatic, but danger for surround people. Infections is often asymptomatic, could be often asymptomatic, sometimes called mild or flu-like of upper respiratory tract disease. It's about all enteroviruses. Immunity. As I told, enteroviruses are cytolytic. Replication happens rapidly and causes direct damage of the target cell and usually massive cell death. Viral shedding from the oropharynx can be detected for a short time before symptoms begin, whereas viral production and shedding from the intestinal may last for 30 days or longer, even in the presence of a humoral immune response. What does it mean? We are not usually protected by the immunoglobulins which produced during the primary immune response, because viral pathogenesis are very fast, and there is no enough immunoglobulins, I mean concentration of immunoglobulins, to protect from the current disease. But in secondary immune response and the secondary infection, it will be very effective. As I told, antibody can protect very well, and it's a major protective immune response to enteroviruses. There is no big role of cellular immune response, but very huge role of the humoral immune response. Secretory antibody can't prevent the initial establishment of the infection in oropharynx and gastrointestinal tract, and serum antibody prevents viremic spread to the target tissue and therefore disease. So both secretory and serum antibodies are important, but if there is no secretory antibody, we can say a person can be infected in primary stage, and during the primary replication in oropharynx and gastrointestinal tract, virus can go out with fishes. So a person which has only serum immunoglobulins can be danger anyway. Cell-mediated immune, as I said, not so important, and its protection role is poor. Enteroviruses, only human pathogens. We can say it's typical anthroponosis. Anthroponosis, human infection. Usually, it causes asymptomatic type of infection. Usually, it also can be found in the environment. I mean, the viruses, because if a lot of people infected by these viruses, we can find a lot of viruses out of the body, so in environment. Usually water can be contaminated. That's why if a person living in the place with poor sanitation and in lack of fresh, clean water, outbreaks sometimes happens. So the outbreaks of enteroviral disease are seen in schools and daycare setting, usually some during the summer. Why the places and why this season? Because schools and daycare sitting places like children garden, because usually children absolutely don't care on the sanitation. Only adult person know and use. Usually you have to wash your hands after visit the toilet. You have to wash your hands before eating something. For children which not under control of the parents, usually it's absolutely doesn't work. That's why it's the places. Why summer? Because during the winter, water much colder and the viruses not so and the viruses can die faster. Okay. For many kind of viruses, it's true, but some viruses, in fact, can be spread through aerosols also, like Coxsackie and echoviruses. Aerosol spreading through the droplets, which a source for which is infected person who are coughing or sneeze, can be important here. So, shortly, about epidemiology. Nature of disease correlates with the specific enteroviruses and age of person. Then ager, then rare infection happens. Infection often asymptomatic with viral shedding. We know that. Viral resistance to environment condition is very high. So detergents, acids, drying, mild sewage treatment, and heat doesn't work to eliminate viruses very effectively. Transmission, mainly fecal oral route with poor hygiene, dirty dippets, and especially their care sitting. Also, possible injection via contaminated water or food. Food, I mean not the food which were cooked properly, for example, fried meat or boiled soup. No, usually food, fresh salad, which washed by contaminated water, or some fruits, which also washed by contaminated water. Usually food should be contaminated through the water source or directly from infected person who don't care on its own hygiene. For example, a chef didn't wash the hands after the toilet and then start to cook shawarma for you. Okay. Hand-to-hand contamination also possible, but rare. Usually it's possible during asymptomatic stage with, let's say, with often contact. Inhalation of infections also possible, but only for Coxsackie and echoviruses, for several serotypes of them. About the group of risk, young children, the main group of risk for polio, for all kind of polio, asymptomatic or mild disease. Children and adults are at risk of polio, also about asymptomatic to paralytic disease, because the paralytic disease is the most dangerous type of clinical syndrome of polioviral infection. Newborns and neonates at high risk for serious Coxsackie and enteroviral diseases. Adult people not very sensitive for such infection. So there are worldwide infections, and we can find all serotypes everywhere, primary everywhere, and usually wild type polio we suggest eradicated in developed countries. but only in developed countries which has the program of vaccination against polio. Right now, there are many countries, perhaps not economically developed, but in this case developed. So they has a program of vaccination. About the summer, I already told. How to control polio? We have several vaccines. So first of all, live oral polio vaccine. It has 3 serotypes and we call it OPV, oral polio vaccine. And also inactivated polio vaccine, we call it IPV. It has same 3 serotypes like oral polio. So only difference here, it's a type of vaccine, live or inactivated. Both of the vaccine are effective. Both of them can be given to the children. There is different way to administrate the vaccine. For oral polio vaccine, okay, through the oral cavity, it's usually drops, oral vaccine, we understand oral. Inactivated vaccine administered through the shots, mean injection. For enteroviruses, for some enteroviruses, we has no vaccination like for all other actually except polio, and only good hygiene and preventive things for people who has Coxsackie and echovirus which transmitted through the aerosols can be effective. About effectiveness of the polio vaccine. Look on the graph. Actually, this graph in United States, because United States started to use polio vaccination very early in history. And they start to use this vaccine actually in the same time, primary. But then oral polio vaccine became danger and they avoid to use it for more than 5 years. After making new live oral polio vaccine, which not danger anymore, they start to use live oral polio vaccine. But killed polio vaccine also still used over the world. And oral polio vaccine also still used over the world. On the graph, you can see total cases number up to 48 cases per 100,000 of population. It's a lot. It means total number. Total number divided to paralytic or non-paralytic polio cases and for only paralytic cases. Paralytic, non-paralytic cases means cases without full paralytic clinical picture, but understandable poliovirus. You can observe only cases with symptoms. Please understand, only symptomatic case. There are many. You would say, okay, 38 cases and approximately 23 of them are paralytic for 100,000 people. So not many. Not many if you have not paralytic polio. Actually many. Because the paralytic polio is not a disease which are recovered. It usually stay with the infected person from all his life. The paralytic polio, very danger. That's why we suggest there are many. So there are several countries, Nigeria, Afghanistan, and Pakistan, when we still have a lot of polio incidents. Primary, why? Of course, vaccination program in some countries, in some, I would say, regions of the countries are work. but in some countries not work, and also different reasons, primary economical reasons, and in some places religion reasons. That's it, actually. We would have about some people who are vaccinated, but has the polio, which get a polio, which results of the mutation of one of 3 strains of polio vaccines. Such kind of cases happened near past, and it's actually very bad, and the primaries happen because of people don't care on the transportation or storage of the vaccine. Usually right now vaccines making pharmaceutical how to say, industry, yeah, pharmaceutical industry is under very strong control from the government systems, usually in developed countries, of course, but transportation and storage can be under control. That's why it's 2 main stage why such kind of problems are happens. Paralytic polio was once considered a middle-class disease because good hygiene would delay exposure of a person to the virus until late childhood. The adolescent years or adulthood, when the infection would produce the most severe symptoms. Infection during early childhood generally results in asymptomatic or very mild disease. Similar to poliovirus infection, Coxsackie-A virus disease is generally more severe in adults than in children. However, Coxsackie-B virus and some of the echoviruses, especially strain 11, can be particularly harmful to infants. I already told, and I will say about once again. So there are several clinical syndromes produced by the enteroviruses, depend on the factor. Which factors? Viral serotype, important to produce specific kind of clinical syndrome. Infecting dose means how many viruses comes simultaneously into the person. Then tissue tropism depend, of course, you know, tissue tropism totally depend on the virus, but we know different tissues has different place and different roads how to come into the tissue. That's why sometimes we need a lot of time, or tissue tropism, of course, affect the symptoms. It's extremely logic. It's if it's infected nervous system, it's neurological problem, skin problem, liver problem. Portal of entry, also important. And we know about 2 main, oral cavity and respiratory system. Patient age, gender, and state of health, of course, important. It's a general information. About the patient age, I already told on the previous slide. And the pregnancy. For pregnant women, some Coxsackie viruses are extremely dangerous. Incubation period for enteroviral diseases varies from 1 to 35 days, depending on the virus. But as you see, it's not very long. The target tissue and the person age also can influence on the period. Then older people for poliovirus, then could be shorter incubation period. Viruses that affect oral and respiratory systems have the shortest incubation period than viral which affect intestinal system. Especially about poliovirus infection. On the picture, you can observe young lady which has paralytic polio for both her legs. And the situation very common in the countries when poor sanitary condition combined with absence of vaccination program. There are 3 poliovirus types, which 85% of the cases of paralytic polio caused by type 1. So type 1 poliovirus, it's a primary causative agent of paralytic polio. Reversion of the attenuated vaccine virus type 2 and 3 to virulence can cause vaccine-associated disease, but not often paralytic polio. And while wild-type polio infection are rare because of the success of the polio vaccines. As noted early, however, vaccine-associated case of polio do occur and some populations remain unvaccinated, putting them at risk for infection. Right now, I would like to tell you about 4 main clinical syndromes of the poliovirus. It's actually only about unvaccinated people, depending on the progression of infection. First type, asymptomatic illness, is the result if the viral infection is limited to the oropharynx and the gut. So virus didn't penetrate into the bloodstream and to the target tissue. So only inside primary cells, primary target cells. At least 90% of all poliovirus infections are asymptomatic. So what does it mean? If in the group of 100 persons, for example, 10 people has some kinds of symptoms of poliovirus, means 90 of all the people has asymptomatic polio. It's amazing result. Asymptomatic means not absolutely not danger. It's danger, but not for a person who are infected with asymptomatic illness. Usually for surround people who are not infected, because transmission is possible from asymptomatic person to health one. Type number 2, abortive poliomyelitis or minor illness, is a non-specific febrile illness occurring in approximately 5 infected persons. Fever, headache, malaise, sore throat, and vomiting occur in such persons due to several days of exposure, usually maximum 7 days, approximately 3 to 4 days. Non-paralytic poliomyelitis or aseptic meningitis occur in 1 to 2% of patients with poliovirus infection. In this disease, the virus progresses into the central nervous system and the meninges, causing back pain and muscle spasm in the additional of symptoms of the minor illness. As you remember, minor illness is a problem of GIT. Such kind of combination can provide a suggestion about non-paralytic poliomyelitis. Paralytic polio, the major illness, occurs in usually less than 1% of infected people, and it's the most severe outcome. It appears 3 to 4 days after the minor illness has subsided, terribly producing a biopsic illness. So means when the first kind of symptoms, I mean the symptoms which related with minor illness like vomiting, diarrhea will gone, several days later, usually paralytic polio are coming. In this disease, wait a second, please. Can you hear me now, Tommy?
Yes, sir.
Okay.
Yes, Professor.
Good. So this major illness, as I said, biphasic. In this disease, the virus spread from the blood to the anterior horn cell of the spinal cord and to the motor cortex of the brain. The severity of paralysis is determined by the extent of the neuronal infection and by which neurons are affected. Spinal paralysis may involve one or more limbs, whereas bulbar or cranial paralysis may involve in combination of cranial nerves and even the medullary respiratory center. What's the problem here? The problem is because of the virus are cytolytic, as you know, means this virus during replication can destroy the cells. The cells of the nervous system, you know, can't replicate fast. That's why primary disease will when destroy a lot of cells, make our muscles without any kind of nervous signals. Or the problem here in the polio. Polio usually can involve several different kind of muscle groups, actually few. It can be one leg or two legs, or there may be completed flaccid paralytic of all four extremities. Sometimes paralytic polio progress over the first few days and may result in complete recovery, residual paralysis, or death. That happens when it's massive paralytic polio and the paralyte a lot of nerve cells. Most recoveries occur within 6 months, but as long as 2 years may be required for complete remission. It's a time when the person has a window for treatment. What does it mean, window for treatment? Window means then early we start to treat paralytic polio, then more chances to get normal. Normal, I mean, not absolutely. In many cases, of course, it's not absolutely, but person would be able to walk without any specific tools or wheelchair or any some cases with there are several cases in the world during when they treated during a lot of therapeutical procedures and physiotherapy, when the person, disabled person, I mean a totally paralytic polio of the child, and the doctor make very nice job and turn back a person to absolutely normal life, so a person could do exercises like absolutely healthy person. I mean playing football, for example. So very danger kind of poliomyelitis, it's a bulbar poliomyelitis, can be more severe, may involve the muscles of the pharynx, vocal cords and respiration, and may result of the 75% death of the patients who has such kind of problem. On the picture you can observe iron lungs. It's like cylinders you can see that operate like a pair of bellows to regulate their breathing and keep them alive. So regulate pressure inside. A person under the pressure and under the high pressure and under the low pressure. And of course, such kind of pressure involve primary respiratory system. You see, there are many people actually absolutely identical wearing. Actually, you can see balloons for children. And of course, it's not normal hospital. It's a primary anniversary, I guess. But there is a true for last century. Right now, of course, we know about many ways how to regulate the respiration without such big boxes, and moreover it's uncomfortable because you need to go to the toilet somehow additionally to the moving. Another kind of polio syndrome, it's a post-polio syndrome. It's not a primary infection, it's a sequela of poliomyelitis that may occur much later in life, 30 to 40 years later than infection first was resolved. In 20 to 80% of the original victims, such kind of polio syndrome could happen. Affected person suffer a deterioration of the originally affected muscle. Poliovirus is not present, but the syndrome is believed to result from a loss of neurons in the initially affected nerves. Such kind of problem usually happen with adult person because nervous system is activity is decreased, the neurons is destroyed by the age processes and can't compensate anymore. As I said, during the primary polioviral infection, neurons are destroyed. That's why it should, in some cases, some destroyed neurons function can be compensated by normal neurons, which healthy. But during the age, normal neurons can't do work for both neurons, means we will die and for its own job. And of course, disease can come back. Please understand, post-polio syndrome not danger because poliovirus are not present in the human body. So there are no viruses totally. Let's make a break for 5 minutes and then I will tell you about Coxsackie viruses and echoviral infection shortly. It will be approximately 10 slides about that.
Okay.
Let's take a break.
Okay, doctor.
Okay.
Okay, doctor.
So let's continue. Coxsackie and Esaulov infections. Tell me, please, can you hear me well?
Yes, sir.
Yes, sir.
Very good. Several clinical syndromes may be caused by either Coxsackie viruses or an echovirus, septic meningitis, by certain illness are specifically associated with Coxsackie viruses. We have two groups. Coxsackie A virus are associated with diseases involving vesicular illness. Hypagny is an example. And Coxsackie B viruses are most frequently associated with myocarditis and pleurodenia. The Coxsackie viruses can also cause a polio-like paralytic disease. The most common result of infection is lack of symptoms or a mild upper respiratory tract or flu-like disease. So usually such kind of infection can be observed as acute respiratory viral infection. The first example, hypagnia, caused by several types of Coxsackie A viruses and is not related to a herpes virus infection. Fever, sore throat, pain on swallowing, anorexia, and vomiting characterize this disease. The classic finding is vesicular ulcerated lesions around the soft palate of uvula. Less typically, the lesions affected the hard palate. The virus can be recovered from the lesions or from face. This disease is self-limited and requires only symptomatic management. There is no specific treatment for it. Look on the picture. It's the characteristic descriptive vesicles are seen on the anterior tonsils pillars. Hypagnia looks like this. It's not a bacterial infection. Another example, hand, foot and mouth disease is a vesicular excitement usually caused by Coxsackie A16 serotype. The name is disruptive because the main features of this infection consist of vesicular lesions of the hands, feet, mouth, and tongue. The patient is mildly febrile and the illness subsided in a few days. Look on the picture, please. Hand, foot, and mouth disease which caused by Coxsackie A16 virus, lesions initially appears in the oral cavity or nearby the oral cavity, and then developed within 1 day on the palms on the picture on soles. Example number 3, and it's about Coxsackie B virus. Pleurodenia or bronchial disease because described there, also known as the devil's grip, devil's grip, is an acute illness in which patients have a sudden onset of fever and unilateral low thoracic pleuritic chest pain that may be excruciating. Abdominal pain and even vomiting may also occur, and muscles hello, can you hear me?
Yes, yes, sir.
Yes, sir.
The message about bad internet connection. Okay, very good, then you hear. If you wouldn't hear me, let me know somehow. Scream, for example. Okay, when I finished. Pleurodenia lasts an average of 4 days, and Coxsackie B virus is the causative agent of this disease. Not all, of course, strains, just several strains. So viral septic meningitis is an acute febrile illness accompanied by headache and signs of meningeal irritation, including nodular rigidity. Pittage or a rash, what you can observe on the picture, may occur in patients with enteroviral meningitis. Recovery is usually uneventful unless the illness is associated with encephalitis or meningoencephalitis or occurs in children younger than 1 year. Outbreaks of picornavirus meningitis of echovirus 11 occurs each year during the summer and autumn, especially in the places especially in the places with very warm summer. On the picture, you can observe the glands which are pressed on the skin, and the rash is not disappear. Instead, it could disappear with other infections. Another enterovirus. For example, Enteroviruses 70 and variant of Coxsackie A24 strain have been associated with the extremely contagious ocular disease infection called subconjunctival hemorrhagic conjunctivitis. The disease has a 24 hours incubation period and will limit 2 or 2 weeks later. Right now, I would like to tell you shortly about the lab diagnosis for all enteroviruses, not only for Coxsackie and echovirus, but also for poliovirus it will fit. So we can do several things. For example, we can use clinical chemistry. What we should do? We should take cerebrospinal fluids from enterovirus septic meningitis can be distinguished from bacterial meningitis, specifically in this sample. Then we can cultivate viruses. For example, poliovirus may be isolated from the patient pharynx during the first few days of illness. From the fishes for as long as 30 days, but only rarely for cerebrospinal fluids. The virus grows well in monkey kidney tissue culture. So, method fit for understanding presence of the viruses in the particular place. Genome and serological studies. Of course, molecular biological investigations like real-time PCR can give us very nice results. But also we can detect specific antibodies in the human serum by using common serological tests, like ELISA, for example. Treatment, prevention, and control. For treatment, some antiviral drugs, pleconazole, is available on the limited basis. The drug inhibits the penetration of picornaviruses into the cell. It must be administered early in the course of the infection. The two types of polioviruses vaccine are, first, as I told you, inactivated polio vaccine, IPV, developed by John Salk, and we sometimes call IPV Salk vaccine. And the second one, live attenuated oral polio vaccine, developed by Albert Sabin. You also can find this name, Sabin vaccine. Both names are equal. Both vaccines incorporate 3 strains of poliovirus. I also already told about it. They are stable, are relatively inexpensive, and induce a protective antibody response. So both vaccines are effective. We should choose what is better, which one is better. And of course, we have to know about each vaccine before using it. That's how to use oral polio vaccine through the oral cavity, just 2 drops of live, as I know, and inactivated polio vaccine also can be injected into the leg, especially for newborn children. So that's it for today. Also, last, what you see here, it's a general information, how and when it should be, but in different countries, there are different ways for the vaccination. Yeah, about Coxsackie viruses, primary, non-specific prevention, hygiene, etc. So, yeah, that's it for today. Fortunately, polio is eradicated in many countries, as I said. In Russia, we have just few cases per year of the polio cases, I mean, incidents, but only in group of people who are not vaccinated only. That's it.
Thank
Have you
you.
any questions? If you have any questions, please be free to ask.